I'm talking about the production of auxin by dying cells. Auxin is a plant hormone. Chemically, it's indole acetic acid. It's related to the amino acid tryptophan and the neurotransmitters tryptamine, 5-hydroxytryptamine or serotonin. This is a subject I've been interested in a very long time. In fact, it was one of the focal points of my PhD research. And I started this line of inquiry around 1964 when I started my PhD in Cambridge. So this goes back a very long way with me. But it's an interest that I've had ever since. And my most recent paper on the subject was published in the Journal of Experimental Botany in 2021. It was a Darwin review called The Production of Auxin by Dying Cells. An earlier paper on this, where I reviewed a lot of the early work on auxin production, showing that it was probably made by dying cells, came out in Biological Reviews in 1973. So you can see this research spans a very long time. When I first got interested in the subject of auxin production, the assumption among my colleagues in plant sciences was that auxin, as an important hormone, must be made in a controlled way under careful feedback biochemical control. And it was assumed it was made in meristematic cells, in other words, young dividing cells in the tips of shoots and roots and in growing leaves. This assumption was very deep-seated, and it's remained the dominant assumption even up until the present. But I think it's misconceived because I think auxin's produced in a completely different way. I think it's produced as a byproduct of cell death, hence the title, The Production of Auxin by Dying Cells. This is surprising, shocking even to some people, but I think the evidence is now overwhelming. When I first got involved in this, it seemed pretty clear that the enzymic steps involved in auxin production involved the degradation or breakdown of the amino acid tryptophan, which is present in practically all proteins. As you see here, the top molecule is tryptophan, and then that's broken down by the removal of its amino group. Amino acids have an amino group, NH2, and this is removed by an enzyme called an amino transferase, and that gives something called indole pyruvic acid, IPA. And indole pyruvic acid is unstable in aqueous solution, particularly at neutral and alkaline pHs, and it breaks down spontaneously to give a range of compounds, one of which is indole acetic acid, auxin. But this process can be speeded up by an oxygenase enzyme that carries out a process called oxidative decarboxylation. And in fact, in plants, these enzymes are very widespread. Well, although all this was known in the 1960s, many people felt this couldn't be the whole story. There must be something much more complicated. And decades have gone on in which people have looked for much more sophisticated, complicated, regulated enzyme pathways, including extremely sophisticated genetic techniques using the model plant Arabidopsis, a small, short-lived plant, thale cress. That's the main model plant used in experimental plant science today. Anyway, after years and years and years of research, finally, the general consensus has emerged that yes, indeed, this is how auxin is normally made. The first enzyme, called TAA, is called tryptophan aminotransferase of Arabidopsis. And the second auxin, YUC, not a very good name for it, comes from a mutant that overproduces auxin in Arabidopsis and makes it look like yucca plants, hence yuc or yucca as the name of this enzyme. It's an oxygenase. Well, this might seem pretty straightforward, and people have worked out where these enzymes are in plants by sophisticated techniques of genetic engineering, sort of engineering fluorescent proteins that light up when the enzymes are there. And they seem to be practically everywhere. And it's usually assumed if the enzymes are there, that must be where auxin's made. But I don't think that's what's happening at all. If you actually look at the affinity of these enzymes for tryptophan and for indole pyruvic acid, they don't have a very high affinity. In biochemistry, there's something called the Michaelis constant that tells you the concentration at which the enzyme is half saturated. And the enzyme, the TAA enzyme, is half saturated at about 300 micrograms per litre. But the amount of the amount of tryptophan in cells is only about 10 micrograms per litre. It's regulated by the tryptophan synthesis system. So there's a low level, and this is what's needed for protein synthesis. The enzyme that puts tryptophan onto transfer RNA for protein synthesis is half saturated at about 5 micrograms per litre. So the amount in the cell is enough for making proteins. It's pretty low level. But the amount that's broken down by TAA and similar enzymes is about 30 times higher. It has to be 30 times the normal concentration before these enzymes swing into action and start making auxin from tryptophan. Now that doesn't happen in normal living cells. Where it does happen is when cells die. Cells are full of proteolytic enzymes which are separated from most of the cell, but when the cell dies they're released and they dissolve the cell. The cell dissolves itself. It's called autolysis, self-dissolving. It happens in animal cells, yeast cells. It happens in all kinds of cells. It's nothing special to plants. Autolysis. So when proteins are broken down, there's about 1,000 times much more amino acids like tryptophan bound up in proteins than in a free form in the cells. So when they're broken down, the levels of tryptophan shoot up. And normally the level would be about here, where the auxin isn't made. It's when the tryptophan levels are here, auxin isn't made. It's enough to make proteins. But when it's 30 times higher, then it starts being broken down by TAA and other enzymes. And although these are usually conceived of as synthetic enzymes, it makes them sound very specific. Actually, TAA, tryptophan aminotransferase of Arabidopsis, is, like other breakdown enzymes for amino acids, pretty non-specific. It works with tyrosine, another amino acid, and 4 or 5 others. It's not even specific to tryptophan. And moreover, the YUC enzyme is a pretty generalized oxidase, monooxidase enzyme that works on hundreds of different substrates, not just indole pyruvic acid. So these are non-specific breakdown enzymes that work on high levels of tryptophan when proteins are dissolving and the levels of tryptophan shoot up. Well, this hypothesis predicts that auxin, indole acetic acid, will be made not just by plant cells when they break down, but by animal cells and by yeast cells. In fact, any cells, as they break down, will release amino acids from proteins. It's pretty non-specific. The proteins are dissolved, releasing the constituent amino acids. There are 20 different ones. Tryptophan is just one of these. And then they're broken down. And it happens that this breakdown product of tryptophan is used as a hormone, a kind of signal of protein breakdown. There are lots of other protein breakdown products that don't have much effect. They're just waste products of protein breakdown. So the first tests I did were to find out whether auxin really is produced by autolyzing cells. And you can kill cells by freezing and thawing them, and then just leave them to break down. And I published a paper on this in Nature, and this shows that auxin is in fact produced by autolyzing plant cells, by yeast cells, and by rat liver cells as they break down. You can see the graphs here. The rat liver produces much more auxin as it breaks down. It's sort of more concentrated. It's got more protein than the plant or the yeast tissue. The axis on the right is referring to the auxin production by autolyzing rat liver. This is all done under antiseptic conditions where there's no bacterial growth, so it's not a matter of bacteria breaking these things down. They're just breaking down under their own autolysis system. And perhaps surprisingly, it's not surprising from the point of view of this hypothesis, but it's surprising from a normal point of view, if you put frozen and thawed sterile rat liver into a plant tissue culture where the cells are just growing as an undifferentiated mass called a callus, it's known that if you put auxin in, a little block of agar containing auxin, it will induce some of the cells to differentiate into xylem or wood cells. So I tried putting in some of this autolyzing rat liver, and sure enough, the rat liver induced wood cells in this plant tissue culture, just like auxin would. So here you see that dying cells can produce auxin. Well, what about in the plant itself? What kinds of cells die in the plant? Well, the first kind that die are the wood cells. All the wood cells in a trunk of a tree, for example, or in the veins of leaves or in a root, are dead, empty tubes. The cells form from a normal cell. They thicken up their wall to make it into a thick tube. They dissolve the end walls so that they're lined up with each other. And then the cells commit suicide. They dissolve themselves by autolysis, creating a tube through which water can pass up from the roots to the shoots. That's what wood cells are. And as they differentiate, as they become wood cells, they die. And most of the sites of auxin production in a growing plant are, in fact, where wood cells are being produced and where they're dying. I've done a lot of work on that. It wasn't known when I started doing this research that wood cells were a place where auxin was made, because most people hadn't looked for that. They just assumed they were made in living, growing, small, young cells. So they'd been ignored. I'm going to do a separate talk on the production of auxin by differentiating wood cells. Another obvious place in which cells are dying is in senescent leaves. Leaves go yellow as the chlorophyll breaks down, and as the leaves senesce, they dissolve themselves. They dissolve the amino acids and the proteins and turn them into amino acids, and then they export the goodies from the leaf down the leaf stem into the rest of the plant so it can be recycled and used for other organs to grow. This transport happens through the phloem, the system of tubes that conduct nutrients and amino acids in plants. So when leaves go yellow, when they senesce, they break down their proteins. So according to this hypothesis, they ought to produce auxin. Well, when I first thought of this, most people said, oh, that's rubbish. They're dying, they can't be producing auxin, it's synthesized by young living cells. So I simply looked at senescent leaf cells to see what happened. And here's a paper I published on the production of auxin by detached leaves as they senesced. This was in Nature. And what this research showed, that as the detached leaves broke down, they indeed produced much more auxin than they had to start with. I then looked through the literature and found that several other people had actually detected this. So it was already there in the literature. It wasn't just me that had found this. And subsequently, this comes from my 1973 review paper, but subsequently there's been a lot more research on this, including in Arabidopsis. And in Arabidopsis, it's now very clear that as the leaves senesce, the levels of free amino acids, including tryptophan, rocket up, and as that happens, they produce auxin. So I don't think there's any doubt now that most senescent leaves accumulate auxin, and this is quite important because auxin helps to delay the abscission, the falling off of senescent leaves. So it'll keep them on as long as the transport systems are working, because the leaves need to pass on their nutrients to the rest of the plant. A few leaves don't seem to show this increase in auxin, and that could be not because they're not making it, but because they also destroy it through oxidation, and if the rate of destruction exceeds the rate of production, they won't accumulate it. But the majority seem to accumulate auxin, just as you'd expect with the dying cell hypothesis. Another place where cells die is in root caps. At the tip of the root, there's something called a root cap, which forms a kind of protective layer. And the cells divide, and then they go up the sides of the roots, protecting the sides of the roots. But then they die. They're there just to protect the root. The inner tissues, which are all living, are protected by this outer layer that's sloughed off and dies. And if you look at root caps, as we see here, the root cap on the right shows cells which have the enzyme markers for dying cells, cells that are dissolving themselves. They're produced right at the tip, and they go on living as they go back, and then they reach a point where they die, and that's the bit that shows up greeny-yellow through these markers of the destruction enzymes. And it's now known that it's exactly there where they produce auxin, and this affects the actual physiology of the root, so it plays an important role, but it's produced, again, not by living cells, but by dying cells. There's a whole lot of other sites of cell death in flowers. In flowers, when the stigmas, when the stamens form, they have anthers, the bits that contain pollen. Those are those little things on little stalks you see in flowers. And the anthers contain a nutritive tissue called the tapetum. The tapetum is made of cells that break down, releasing nutrients that feed the growing pollen grains. And you see here, using a special stain that shows the presence of auxin through auxin responder proteins, you can see on the left a flower where the very heavy stain in the anthers shows a very large amount of auxin, and on the right you can see blue stained cells, the tapetum. The grey ones are the pollen grains. Those are breaking down and contain auxin as they break down. As the flower develops, the stigma, the bit that sticks up that the pollen grain lands on, has a lot of little hairs on it called papillae, and these papillae break down, and here you see the staining that shows the presence of auxin. On the left you can see the young papillae which have just started to break down. They're intensely releasing auxin. Rather older ones on the right. You can see the pollen containing anthers below that, which are also rich in auxin, as I've just said. Then, as the pollen grain moves down the stigma, cells die, and again auxin is produced as that happens. Then the seed forms, and in the seed there's a nutritive tissue called the nucellus, which is then followed by another nutritive tissue called the endosperm, which store goodies for the developing seed. When a seed germinates, these nutritive tissues break down to release the stored goodies that the young developing seedling, the embryo, uses to grow. Well, when the nucellus breaks down, there's a burst of auxin production, and similarly, when the endosperm breaks down, there's yet more auxin production. Germinating seeds are very rich in auxin as these nutritive tissues break down. Again, there's now a great deal of evidence for this. Auxin is also produced when cells are wounded. If you cut a plant, for example, if you cut a potato in half, you wound the cells that you cut, and they break down, and they produce auxin. The first research on this, actually, which was what got me interested in the subject in the first place, was done in the 1910s and 20s by a German botanist called Haberlandt. And Haberlandt showed that if you cut potatoes in half and you then look what happens, the cells underneath the cut start dividing to seal off the wound. It's called the wound response. And the wound response depends on chemicals released by the dying cells. Haberlandt found that if you wash the surface, the cut surface, so you wash off all the bits released by the dying cells and wash away the contents of the dying cells, the cell division doesn't happen. If you get extra bits of dying cells, if you pulp up potato, kill lots of cells and add more on the top, you get much more cell division. And more recent research has shown that yes, indeed, during this process auxin is produced, and it's the auxin that stimulates the cell division. So it's now known that dying cells produce it as well. In fact, practically all the places in the plant where auxin is made are sites of cell death, which is why I think that the dying cell hypothesis is enough to explain almost all auxin production. However, there's one exception to all this, which here's a picture of the wounded cells. Underneath a wounded cell on the potato, the cells that divide, forming a kind of corky layer to seal off the wound. There's one exception to this, which anyone who's studied botany will probably think of, which is the famous example of coleoptile tips. In grass seedlings, for example, oat seedlings, the primary shoot has a sheath around it called a coleoptile. The first leaf is inside this sheath. And Charles Darwin, with his son Francis, discovered that the tip of the coleoptile is sensitive to light and affects the way the plant grows. As you see here, this as a textbook diagram for the discovery of auxin. When exposed to the light, it will bend towards the light. If you cut off the coleoptile tip, that doesn't happen. If you put a hood over it, a dark hood, it doesn't happen. If you put a transparent hood over it, it does happen. If you put a dark hood, a dark barrier around the base, it doesn't stop it bending. So the tip is light sensitive, and something about it causes the cells below to grow, and they grow more on the side away from the light than on the side with the light, which causes it to bend. Now, the early discovery of auxin was first established in the 1920s through research with coleoptile tips. If you cut the tip off, it stops growing, showing that the tip is producing auxin, or at least releasing auxin, that causing the rest to grow. If you put the tip on an agar block and then put the agar block on the cut stump, it will grow, showing there's something in the agar released by the tip that's making it grow. If you put the agar block on one side rather than the other, so it's asymmetrical, it will grow asymmetrically. It will grow more on the side where you put the agar block and bend as it does towards the light. This is classic work on auxin that first triggered off interest in the hormone, and its role in cell extension underlies the phototropic and gravitropic responses of plants, the way that shoots bend towards the light and against gravity, going straight up, the way roots bend away from the light and in darkness grow straight down, the primary roots grow straight down. All of this is mediated by auxin and differential auxin transport. So thinking about auxin was heavily influenced by this work on coleoptiles, and people assumed the auxin was synthesized in the coleoptile tip. Well, there aren't any dying cells there, so it didn't fit my own hypothesis. This was the one exception that didn't fit. So I looked again at the research on coleoptile tips. I published a paper re-examining the evidence that supposedly showed auxin was produced by coleoptile tips. And what I found was that it doesn't get produced in the tips. Instead, the auxin is coming up the xylem, up the sap, with the water up the wood tubes, from the seed where the endosperm, the storage tissues, are breaking down. It's full of auxin. It's being carried up in the sap and it accumulates at the tip. And it's not just auxin that accumulates at tips. Any substance that's in the xylem will accumulate. It gets more concentrated, it evaporates from all over the plant, but leaving what's inside the xylem getting more and more concentrated to the tip. If you do experiments with dyes, this was done with acid fuchsin, there you see how the auxin on the top left is accumulating at the tips of coleoptiles. The bottom left, it's accumulating at the tips of primary leaves. It's not just specific to coleoptiles, it happens to leaves. On the bottom right, you see how it happens in the tips of petals as well. This is a perfectly general process. And I showed with radioactive indole acetic acid, radioactive auxin, that exactly the same thing happens with auxin. It's just harder to see. In my paper, I give the results of the radioactive experiments, but you can see it very easily with the dye. Now, one of the things that was known from this classic work on auxin was that if you cut the tip off, after an hour or two, the plant starts growing again, and the tip now contains auxin, and it's called the regeneration of the physiological tip. And so people assumed that somehow the cells had started synthesizing auxin, but actually that's not the case. As you see in the top right of this diagram, the dye accumulates at the cut stump. So if you cut the tip off and there's auxin constantly coming up, it now accumulates at the stump. And again, you have a source of auxin at the tip moving down, behaving as if the auxin's made at the tip, but it isn't. So this seemed to be the one exception that refuted the dying cell theory of auxin, and it turns out not to be an exception at all, because the auxin involved in coleoptiles isn't made there, but it's carried there from the seed, where it comes from degenerating nutritive tissues. Now, there is one other exception that seems to show that auxin can be made by living cells. In fact, it does show that auxin can be made by living cells, but it's the exception that proves the rule. And this is from a thing called crown gall. Crown gall is a kind of gall, a cancerous growth in plants. You see one here on the stem, which is caused by a bacterium called Agrobacterium tumefaciens. And it was, in fact, I started my research with Agrobacterium tumefaciens. That was my very first experiment. I started looking into how it worked and how it infected plants. And that's what got me interested in wounding, because it will only cause a cancerous growth if it gets in through a wound. And it's now known that what this agrobacterium does is genetically modifies the plant cells. It contains a plasmid which inserts genes into the plant chromosome, genetically modifying them. And what it does is inserts enzymes that really do synthesize auxin. And these cells make their own auxin, which is why they escape from the normal controls of the plant, because making their own auxin, they can just go on and on dividing, and they become cancerous. Incidentally, Agrobacterium tumefaciens, with its ability to genetically modify plants naturally, is used as a carrier for genetic modification. So when you read about GM crops done with Roundup Ready to make Roundup Ready soybean, for example, they're using this cancer-producing bacterium to introduce the genes into plants. It doesn't cause cancer of the plant when it's used for genetic modification, but they're using this system that the bacterium has evolved over the century, over the millennia, over the millions of years, as a way of subverting the plant by genetically modifying it. And what it does is introduces two new enzymes. It introduces several new enzymes, but the two that concern us here are an enzyme called tryptophan monooxygenase, which, as on this diagram, you can see that it turns tryptophan by removing the acid group, the CO2H group. It turns it into something called indolacetamide, which is what you see in the middle there at the left. On the right is the normal pathway of auxin from tryptophan through indopyruvic acid to indolacetic acid, auxin, IAA. But this route in the crown gall involves tryptophan monooxygenase producing indolacetamide, and that is then oxidized by indolacetamide hydrolase, which turns it into indolacetic acid. So these are special enzymes the plant doesn't normally have. Unlike the tryptophan aminotransferase, which has a Michaelis constant of about 300 micrograms per litre, very high Michaelis constant compared with the 10 micrograms per litre that's present in the plant cells. This enzyme, the tryptophan monooxygenase that produces indolacetamide, is active at 10 micrograms per litre. That's its Km, its Michaelis constant. It's half saturated at exactly the concentration found inside plant cells. So in a normal plant cell without cell death, it does produce indolacetamide, and then the intermediate, the next enzyme from indolacetamide has a Michaelis constant of 1 microgram per litre. And so this enzyme is able to break it down and make IAA. So the whole of this process can take place in living cells. But as I say, this only happens in genetically modified living cells. It doesn't happen in normal plant cells. They only produce auxin when they die. So I think this gives us a completely different way of thinking about plants. It shows that death is part of life, that the dying of cells within a plant is actually giving more growth. More growth gives more differentiation of wood cells and more cells that die, and that gives more auxin that gives yet more growth. There's a positive feedback, which is why plants keep growing unless they go dormant. And the dying cell hypothesis accounts for practically all the facts that you need to know about how auxin's made in plants. If you want the references for all of this and want to read about it in more detail, do please go to my most recent paper, the one in the Journal of Experimental Botany, which you see here, The Production of Auxin by Dying Cells. There's a full bibliography, and you can find all the references and all the details there. The links are given below this YouTube. I'm giving another talk on the production of auxin by differentiating wood cells, and I'm also doing one on the evolution of auxin. How did auxin evolve as a plant hormone? I have other talks on how auxin is transported in plants, how it affects plant polarity in my talk, The Polarity of Plants, and how it's transported from the shoots towards the root tips by the polar auxin transport system. That talk's called Polar Auxin Transport. So this is part of a series of talks on auxin, one of the scientific topics that's occupied me scientifically practically the whole of my scientific career. So I'm very pleased to have this chance to summarize some of it here.